Good afternoon to everybody, all respected executives of the NPT and dear participants. Last week, we have completed 2 sessions of GCP on overview of GCP and responsibilities of stakeholders in clinical trials. I think NPT has also circulated the PPT as well as video recording of the event. My sincere request to all participants that if anybody wants to ask any question, then they can post the question in chat box. We will take it during the discussion or will be addressed by the speaker in chat box also. Today, we are having 2 new topics. First is on clinical trial protocol, investigator brochure and essential documents, which will be taken by Dr. Bhagwat Gunale sir. And the second session is on informed consent process, which will be taken by Dr. Hira Bhalla sir. The first session will be moderated by Dr. Sunil Mahakarkar sir. Sir is presently working as professor and head Department of Pharmacology at GMC Nagpur, Maharashtra. Sir has 15 national and 2 international publications in Index Journal. Sir has also worked as faculty for Regional Center for Medical Education Technology Unit, MOHS Nasik, and Good Clinical Practice Workshop at GMC Nagpur. He holds a PG Diploma in Bioethics and has worked as Member Secretary, IEC, IGGMC Nagpur, and as a member in IEC, GMC Nagpur. Sir has also presented one paper titled Levamisole as an immunomodulator in fresh pulmonary tuberculosis and was awarded travel grant by British Columbia Lung Association. I welcome you, sir, for the session.
Thank you.
The next session will be moderated by, second session that is moderated by Dr. Samina Farhat madam, who is presently working as professor and head Department of Pharmacology, GMC Srinagar, Kashmir. Madam is also chairperson of Animal Ethics.
My voice lost.
Sir, your voice is lost. We can't hear you.
I think his internet is disconnected. He has to join back.
आपको दिख रही है? Am I audible, sir?
Yeah.
Yes, sir.
We are ready.
Yes, sir.
Okay. Just a minute, sir. So Dr. Farhat madam is presently working as professor and head pharmacology GMC Srinagar. Madam is also chairperson of Animal Ethics Committee and member of IEC GMC Srinagar. She is also chairperson Jammu Kashmir Medical Sales Corporation and member of Ethics Committee Schemes SOVRA. Madam is also in charge of Chief Librarian GMC as well as associated hospitals. And Madam has published 50 publications in national and international journals. She has also authored practical notebook of undergraduate students as per CBME curriculum. I also welcome you, madam, for the session.
Thank you, sir. Thank you so much.
Now I request Dr. Mahakarkar sir to introduce today's first speaker, Dr. Bhagwat Gunale sir. First session will be of 20 minutes, followed by question and answer session for 10 minutes. Over to Mahakalkar sir.
Thank you, Dr. Dakre, sir. I am privileged to introduce Dr. Bhagwat Gunale, who will be speaking on clinical trial protocol, investigator brochure, and essential documents. Dr. Gunale has done his MD from CMC Vellore, and at present he is working as Deputy General Manager, Clinical Research and Pharmacovigilance at Serum Institute of India. He is a clinical pharmacologist by training. He has 15 years of experience in pharmacology industry, clinical development, pharmacovigilance, medical affairs, clinical regulatory for various vaccine and biologicals. Also, he has got 18 publications in peer-reviewed international journals. So, we are privileged today to have a talk on clinical trial protocol by Dr. Bhagwat Gunale. Here I present him as the presenter. Thank you.
Thank you, sir, for the kind introduction. I'm just opening my PPT. So I hope you can see the screen.
Yes, sir. Yes, sir.
So I will be talking about clinical trial protocol, investigators brochure, and essential documents in line with the ICH GCP guidelines, as well as the Indian new drugs and clinical trial rules specific requirements. Before going into the ICH GCP protocol and IB, I will just include some historical background, then the sections of ICH GCP guidelines, compliance with the protocol, and then the sections of protocol and IB as per the ICH GCP guidelines and essential documents. So before ICH GCP guidelines came into effect in 1995-96, there were various guidelines basically directing the basic ethical principles to be followed in research, which always started with some controversies with the human experimentation. So in 1931, there was the ethical guidelines for biomedical research. After this, the Nazi physicians committed some experiments on the war prisoners, and which were later on, after Germany lost the war, these crimes were detected and then they were tried in the court of law. And then after this, the Nuremberg Code came into effect, which was basically to ensure that the experiments are done with the voluntary consent of the participants. So the concept of voluntary consent came into effect with Nuremberg Code. And then some anomalies caused by the thalidomide across the world in Europe, Canada, led to the Kefauver amendments in the Food and Drug Cosmetic Act in USA, which was passed as a law in 1962 in USA. Later on, there was a Declaration of Helsinki, again, basically guiding the ethical principles in research. And this Declaration of Helsinki is updated from time to time. And currently now it has 35 principles, which was latest modified in 2013. And then there was a Belmont report, which was written in US, again guiding the ethical principles for biomedical research involving the human subjects. Then the ICH GCP guidelines, the first version was approved in 1995 and came into effect in 96. That was E6. And then now E6R2, which came into effect in November 2016, which we are currently following. So these ICH GCP guidelines has various sections: glossary, then principles of GCP, requirements of IRB, IEC, responsibilities of the sponsor, then the responsibilities of the investigator. So all these sections are already discussed in the previous session. And now I'll be focusing on the clinical trial protocol and investigators brochure and essential documents. Whenever a trial is conducted, it has a protocol, and all the stakeholders who are involved in the conduct of the trial, they need to be compliant with the protocol. And if there are any deviations from this approved protocol, then this should be documented appropriately and notified to the sponsor, ethics committees, and the regulatory authorities as appropriate. In certain cases, the amendments may be implemented to avoid immediate hazard or immediate safety issues in the participants. In the interest of protection of the subjects or the participants, sometimes the amendment may be implemented without the approval of IRB, sponsor, and regulators, but that should be informed to all these stakeholders as soon as possible. And so I have already covered this. So now coming to the clinical trial protocol and protocol amendments. So this is the template of the protocol, the sections of the protocol as per the GCP. The first section is general information, then the background, objectives and purpose of the trial, trial design, selection and withdrawal, that is eligibility criteria, any discontinuation criteria, what kind of treatment regime would be used, and then the assessment of efficacy, safety, the statistical considerations in the trial, and then the source document, access, the quality maintenance in the trial, the ethical considerations, data handling and record keeping, the finance aspect of the trial, who would bear the expenses for the trial, how would be the medical care of the subjects be taken, insurance aspects as per the regulatory requirements in particular region where the trial is being conducted, the publication policy, and any other supplementary documents. So now the first section is the protocol title. So the study should have a proper title identifying the investigational agent, the brief of study design, the population that will be included. Then there would be a protocol acronym or a secondary protocol identifier and the date of this protocol. And if the protocol underwent any amendments, then those amendment numbers should also be there along with the date. Though this title page does not have IND or investigational product name as per ICGCP, but as per NDCT, there should be the name of the investigational product on the title section. And the name and address of the sponsor and the CRO which is conducting the study. Name and title of the person who is authorized to sign the protocol for the sponsor. Then the name, title,
Sir,
and
your
address.
voice is low. Voice is little bit low. Can you speak a bit louder?
Yeah. So the name, title, and address, telephone number of the sponsor's medical expert, if this is a person different from the signatory for the protocol. This is better now, sir? Then the name and title of the investigator conducting the trial, and address, telephone number, as well as all the participating sites, physicians or the investigators, along with their address. And then the clinical laboratories involved in the conduct of the trial. So these can be the laboratories which are doing the safety laboratory testing, like the biochemistry, hematology, or any other special assays. And in case of vaccine trials, there would be the laboratories which are analyzing the immune responses in the form of antibody titers. And any other collaborators or technical departments which are involved in the trial. Sometimes there can be a sponsor and co-sponsor, or there can be some technical collaborators, or there can be a sponsor and an additional funding agency. And then the background information. In the background information, we need to include the name and the description of the investigational product, summary of findings from the clinical studies as well as the non-clinical studies which are relevant to that particular clinical trial in the humans. Depending on the phase of the clinical trial, the amount of data would vary. Suppose if it is a phase 1 trial, then it would have only the non-clinical data for that product. But if it is a phase 3 trial, then it would have a relevant phase 1 and phase 2 clinical trial data. And if a particular drug is from a therapeutic class where there are already approved drugs, then some background about those drugs or the available treatment in that domain should be included. and how this new treatment adds to the therapeutic armament. And then the summary of known and potential risks and benefits. Again, if it is a phase 1 trial, then for that particular drug, you may not know the potential risks and benefits, but from that therapeutic class and based on the pharmacological class of that drug or the intended action, you would have those known potential risks and benefits. And then the justification for the route of administration, the dose and the dosage regime, that is either a single dose or a repeated dose, the duration of the dosing, and the treatment periods. The treatment periods means sometimes there are crossover trials using the same participants with a washout period in between, or there is a parallel design study protocol where you give the treatment over a period of 3 months and observe them for 6 months or 1 year, depending on the protocol design. Then there should be a statement that the trial will be conducted in compliance with the protocol, GCP, and applicable regulatory requirements in that particular country where the trial is being conducted. Description of the population to be studied in brief, that is either the pediatric population, adults 18 to 60 years, elderly population, healthy or diseased, depending upon the indication or the drug that is being studied in the clinical trials. And then the references to the literature and data that are relevant to the trial. As I mentioned, that if this is a novel drug, suppose for a pulmonary tuberculosis, then you have to give some references to the already existing literature about the existing treatments and what is this trial going to add, or the rationale, basically the rationale for the trial that this new drug would overcome the limitations of multi-drug resistant tuberculosis or extremely drug-resistant tuberculosis. Likewise, so it would depend on the category of the therapeutic. So for each class, these references to the literature and data would differ. Then the section of the trial design and endpoints. So after that background general information and the background information in the therapeutic area and the existing information about the drug, you have to explain the objectives of the study. So the objectives will be primary objectives, secondary objectives, and exploratory objectives. So the objectives of the trial would depend on the phase of the trial. If it is a phase 1 trial, then the primary objective would be safety. And if it is a phase 2 trial, then the primary objective will change to safety as well as the effective dose. So safety and efficacy will become primary or co-primary objectives in the phase 2 trials. But again, the importance is for the safety in the phase 2 trials, because phase 1 trials are done in healthy population, healthy adults. The phase 2 trials are done in patient population or the target age group. So in case of vaccines, it is most of the times healthy population all across, or those with the controlled comorbid conditions. And in phase 3, the difference between the various phases and the objectives is the difference in the sample size. The phase 1 trials are generally conducted in less than 100 individuals. The phase 2 trials are conducted in 50 to 200 to 400 individuals. And the phase 3 trials are generally conducted in thousands of individuals. So the objectives and the purpose of the trial would be described in this section. And then the design. So the clinical trial design. So the scientific integrity of the trial and credibility of the data from the trial depends substantially on the trial design. So the trial design should include the primary endpoints and the secondary endpoints. So the endpoints again can be the safety in terms of adverse events or serious adverse events, or the efficacy endpoints, the efficacy of the drug or the comparator product, or if there is no treatment in that domain, then the efficacy in comparison to the placebo, or the currently given standard of care supportive symptomatic treatment. And then the secondary endpoints would be additional So in case of phase 1 trial, the secondary endpoints could be the pharmacokinetics of the drug, and the primary endpoint would be the safety. So in case of phase 2 and phase 3 trials, the primary endpoints would be again, for phase 2 trials, the primary endpoint will be again safety. And in phase 3 trials, the primary endpoint would be the efficacy. So efficacy, again, efficacy or effectiveness, depending upon the therapeutic area. And then the description of the type of the trial or design of the trial, like a single blind or open label or double blind, placebo controlled or active controlled. Whenever there is a treatment which is approved for a particular condition, then the placebo controlled trials are not feasible in such indications. Then you have to use the standard of care or the approved treatment as a comparator. And then the design can be a parallel design or a crossover design. A parallel design means you have two treatment arms. One would be your investigational product, and the other comparator group would be the comparator product or a placebo. And this section also should show the schematic flow or the flow chart of the study design. Then the protocol should explain the measures which are taken to minimize or avoid bias, that is blinding, that is double blind, single blind, or observer blind, depending upon the investigational product that we give. So blinding may not be possible in cases where the intervention that you administer is different. So in such cases, you may need to have two teams. One is a randomization and allocation of the treatment and administration of the injection or the tablets to the participants. And the blinded team would be involved in monitoring the study outcomes, that is the safety outcomes and efficacy outcomes. So this should be explained in detail in the protocol as well as any other plans that are developed as addendum to the protocol. And the procedure for randomization, whether it is a paper-based randomization or it is a electronic system-derived, that the interactive voice response system randomization or interactive web response system-based randomization. As well as you need to describe the treatment, that is a single dose or a multiple dose, the duration of the treatment, the dose, the form of the dose, that is either it is a tablet, capsule, or it is an injection, it is a route of administration, whether it is an intramuscular injection, intravenous injection, or an infusion, or a local application on the skin, or inhalation. So that should be explained in the detail, and how the products would be packaged and labeled, whether the packaged products or the labels would be identical for the investigational product and comparator product to maintain the blinding, and that would decide whether you need a separate blinded team or unblinded team, or all the stakeholders will be blinded if you have identical packaging. Then the duration of the participation for each subject in the trial, it can range anywhere between 30 days to or several years, depending upon the nature of the product and the outcomes that are intended to be monitored in the study. And then the description of the sequence and duration of the trial. Where sequence means if it is a crossover trial, then there could be half of the participant receiving the investigational product first and the comparator product after a washout period, or the half of the subjects receiving the comparator product first and the investigational product later, with a washout period depending upon the pharmacokinetics and pharmacodynamics of the study product. Generally, the crossover trial designs are used in the BA/BE studies and not in the clinical trials. And the duration of all the trial periods, that is the treatment, there could be a variation between the treatment period and the follow-up period. Sometimes the treatment is given for 3 months, and then the participants could be followed for 6 months, 1 year for safety or efficacy. Then the stopping rules or the pause criteria for the trial in case of there is an increased number of serious adverse events or increased number of severe adverse events, or there are any other unusual adverse events which create a safety concern for the participants. So based on that, the stopping rules or the pause criteria or the discontinuation criteria for individual subjects for a particular part of the trial. A trial may be conducted in 1 or 2 parts, and the entire trial should be defined in the protocol. Besides this, the accountability of the investigational product, the procedure which will be followed for the accountability, that is how many vials, tablets or injections were supplied to the site, and how many were used, how many were unused, how many were returned by the participants, whether they were verified by the monitors, and whether they were destroyed after the verification. All this should be documented in the protocol. Then the maintenance of the trial treatment randomization code, how the access control would be maintained for the randomization code, especially if it is a paper-based randomization or electronic system where there should be access control, that the investigator and team, as well as the sponsors, don't know in advance that what treatment will be received by the next participants. So this will avoid the bias, as well as in case of any serious adverse events, if the investigator needs to know the treatment code for the management of the adverse event, then there should be a procedure for breaking the code, or we call it as a unblinding procedure. Or during the COVID-19 vaccine clinical trials, the emergency use authorization was granted for the vaccines. So during such periods, you could no longer maintain the participants in the placebo arm on the placebo. So after a certain period of safety follow-up was completed and the vaccines were available, they were needed to be unblinded. So you needed to define the procedures for unblinding, and also to maintain that safety follow-up without introducing any additional bias.
Sir,
And
go little bit fast as time is less, sir.
Yeah, identification of any data to be recorded on the CRFs. Then the inclusion criteria and exclusion criteria you need to define beforehand. The withdrawal criteria, if during the enrollment, if there was non-compliance to these inclusion or exclusion criteria, or any inclusion exclusion criteria were met after the randomization, then you need to define when and how to withdraw the subjects from the trial. The type and timing of the data to be collected on withdrawn subjects, whether they will be included in analysis or not, that should be defined in the protocol as well as SAP, and the follow procedure for the withdrawal of the subjects. The treatment names, as I mentioned, should be the treatment details should be described in details, and procedure for monitoring subject compliance. This is especially important for the repeated dose treatments, especially the oral treatments, or wherever the subjects take the treatment at home. Then the assessment of efficacy parameters should be defined, and the methods and timing of assessment of efficacy over a certain period, as well as how these efficacy assessments will be recorded and analyzed. Assessment of safety, that is specification of safety parameters, which can be laboratory changes, which can be adverse events of special interest, SAEs. The methods and timing for assessing and recording these parameters, as well as analyzing these parameters. And then the procedures for eliciting these reports and reporting these events, and the duration of follow for these events. Then the statistics section needs to define the statistical methods to be employed, including the timing of analysis, as well as any planned interim analysis, number of subjects planned to be enrolled, that is sample size calculation and justification for that. In multicentric trials, then the projected number of subjects to be enrolled at each site should be defined, or that should be clarified that this does not affect the trial outcome if only a few sites enroll out of all the selected sites. And then the justification for the control group should also be included, and the sample size justification. The criteria for termination of the trial, if you find any unusual side effects or something in the trial, or there is no benefit seen from the interim analysis. Procedure for reporting deviations from the original statistical analysis plan, and selection of subjects to be included in the panel, that is per protocol analysis population, ITT population, that is intention to treat population. The protocol should define that the sponsor should have that access to all the source documents, and the sites should allow access for independent audits, monitoring, IRB, IEC review, regulatory authority inspections, and they should give access to the source document. The quality control and quality assurance aspect should be mentioned in the protocol. The ethical considerations like audiovisual recording of the consent as applicable for that particular age group, the assent, consent, all that should be described. And the financing and insurance aspects, the medical care of the participants for related and unrelated adverse events should be defined. And publication aspects of the trial should also be described. So coming to the investigator brochure, investigator's brochure is a compilation of clinical and non-clinical data that are relevant to the evaluation of the study products in the clinical trials. The investigator and others who are involved in the conduct of the trials need to understand the rationale for the study and to maintain the compliance with the key features of the protocol like dose frequency, the safety, all that they need to understand, and for that investigator's brochure is essential. It provides an insights to support the clinical management of the subjects during the course of clinical trial. The information in an IB should be presented in a concise, simple, objective, and balanced non-promotional form, which should enable the clinician or the investigator to understand it and make his or her own unbiased risk-benefit assessment, that whether the risks and benefits, that the risks are less than the benefits, and any adverse events, if they occur, then how they would manage, all that they will understand by reading the IB. In case of licensed products, IB is not prepared generally, and the package insert or the summary of product characteristics is used as a investigator's brochure. And for the new products, the new IB, the IB is prepared, which has different sections. So this is the title page for the IB, which is already there, and there are 6 sections. So summary, which highlights the crux of all these 2, 3, 4, 5, 6. That is summarizing the significant physical, chemistry, pharmacological, toxicological, and clinical information, depending on the stage of clinical development. So introduction section will talk about the disease and the available treatments in that therapeutic area. Then the physical, chemical, and pharmacological properties, that is CMC, what we call the chemistry, manufacturing, and pharmaceutical dosage formulation related information. And then the non-clinical toxicology, non-clinical pharmacology, and toxicokinetics data. And then the effects in humans. So if it is a phase 1 study, then we will not have any effect in human for that particular product, but we will have the data coming from the other therapeutic agents approved in that class. So when the data is available, it would be pharmacokinetic data, the safety efficacy data, and for phase 3 trials, the marketing experience will not be applicable. But if it is a phase 4 trial, then the marketing experience from that product or from the comparator products in that therapeutic area should be included in this. And then the summary of data and guidance for investigator, especially with respect to the administration of the product, any safety concerns should be highlighted in this. So I'll skip this. Now, essential documents. So essential documents are defined as individual. Basically, these will permit individually and collectively the evaluation of the conduct of the trial and the quality of the data produced. They serve to demonstrate that the investigator, the sponsor, and the CRO or the monitors complied with all the standards of the GCP, and that gives evidence that the data are credible and it meets all the regulatory requirements. And these are usually audited by the sponsor's independent audit function or by the regulatory authorities, so as to confirm that the trial was conducted as per the applicable GCP guidelines and there was no compromise in the integrity of the data collected. And these essential documents are grouped depending on the phase of the trial, before the phase of the trial starts, during the conduct of the trial, and after the completion or termination of the trial. And basically all these documents are archived in a trial master files, which are established at the beginning of the trial, both at the investigator's location, that is at each study site, as well as with the sponsor, and the sponsor sometimes can delegate this activity to the CROs which are conducting the study. And this trial master file should be updated from time to time during the course of the study. So when a final closeout for the trial is done, at that time, this file should be reviewed by both the investigator and the sponsor to ensure that all the documents are there in the files. So if any of these documents are missing in the files, then this is considered as a serious deviation, and then the investigator as well as the sponsors become liable for the action by the regulatory authorities. So this checklist is already easily available in the GCP document, so I will not go through this. Thank you.
Thank you, Dr. Gulade. Sir, there are a few questions. The first question is the placebo-controlled trial ethical?
So placebo-controlled trial would be allowed when there is no treatment for that condition, no approved treatment for that condition. For example, when we initiated the COVID-19 vaccine trials in 2020, when there was no other vaccine approved, at that time point, the placebo-controlled trials are acceptable. Suppose now, for example, there is now, say, respiratory syncytial virus vaccine. So now currently there is no approved respiratory syncytial virus vaccine available globally. And even if it is available in United States and it is not available in India, and this vaccine is not part of our national immunization schedule, in such cases, you can do the placebo-controlled trial. But if your country's either essential drug list or your national protocols for the treatment of that particular condition or prevention of that condition has a recommended treatment or a preventive measure, in that case, you will not be able to use placebo control. But whenever there is no such treatment available, then the placebo control trials are acceptable.
The second one is what is exploratory objective?
Basically, exploratory objectives are like additional objectives which may be done, may not be done. They are not mandatory per se, but the primary objectives and secondary objectives are mandatory. And generally, the primary objective is a statistical mandate, especially for the phase 3 trials.
Thank you, sir, for your elaborate deliberation. Now I hand over the session to Dr. Dakle, sir. Thank you.
Thank you, Dr. Gunale, sir, as well as moderator Dr. Mahakalkar, sir. Now I request Dr. Samina Farhat, madam, to introduce today's second speaker, Dr. Hira Bhalla, sir. Again, second session will be of 20 minutes, followed by question and answer session for 10 minutes. Over to Farhat, madam.
Thank you, sir. Thank you so much. The second speaker for today's program is Dr. Hira Bhalla, and I am very privileged and honored to introduce him. He is presently working as an additional professor of pharmacology, AIIMS Gorakhpur. He is a member of the subject expert committee of CDCSO DCG Delhi. He has completed some extramural funded projects with ICMR and ANRI and COVID-19 vaccine. He is the principal investigator in 3 regulatory trials and co-PI in 6 trials. And he has 21 publications in international and national journals, including Lancet. And he has published some books for self-assessment and review pharmacology. And he's the coordinator for PVPI and joint member secretary at AIIMS Gorakhpur. And he's the nodal officer for COVID-19 vaccination drive for AIIMS Gorakhpur. Welcome you, sir. Please start your presentation.
Thank you, ma'am, for your kind introduction. Sir, I am audible?
Yes, sir. Yes, sir, you are.
Is my screen shared?
Yes, yes.
Yes.
It is shared, but make it full.
Okay, sir. Okay, sir. Sir, it is now in slide mode.
No, sir, no.
Sir, just wait, sir.
बल्ला सर उसको एक बार क्लोज करके दोबारा करके देखिए सर.
हाँ सर, हो जाएगा 1 मिनट सर.
Yes. Just go on view. Balla.
Yes, sir. 1 minute, sir.
Yes.
Slide show नहीं, just wait.
व्यू में गए तो हो जाएगा सर.
View में
व्यू टास्क बार में लिखा हुआ है व्यू, वहां पे.
बार में व्यू सर 1 मिनट.
होम के लाइन में है सर. 8th और 9th टैब.
The slide show.
एक मिनट. हाँ. ये
एक बार बंद करके बोलता हूँ.
ये तो है. ये कर दो. स्क्रीन कर दी. हाँ करे. हाँ शेयर. हाँ करिए. हाँ युवा स्क्रीन शेयरिंग आपकी दिख रही है. नहीं, फुल स्क्रीन नहीं हो रहा. अच्छा.
हाँ. Yes, yes, correct.
बिगिनिंग से करना.
हाँ सर, हो गया
हाँ,
सर.
जम गया.
हाँ हाँ.
Now you can go ahead.
Yes, sir. First of all, namaskar to all. Thank you, NPT, National Association of Pharmacology and Therapeutics, and Department of Pharmacology from AIIMS Nagpur, to giving me opportunity to deliver a talk on informed consent process. As discussed by our previous speakers, Dr. Dennis, sir, the foremost challenge for conducting clinical trial is properly obtained informed consent process, sir. So informed consent process or informed consent is the only part which making a research ethical, sir. So most of my slide is from my previous experience. And overview of my presentation, including how to inform or participant understanding, informed consent process, including participant information sheet and informed consent form, what to inform, that is elements of PIS, essential steps of conducting, whom to inform, and different type of consents. So first of all, understand the consent process. Informed consent जितना भी हमने अभी तक 9 ट्रायल्स में मैंने किए हैं, वो उसको अंडरस्टैंड करने के लिए एक हिंदी में मैं उसका आपको दिखा रहा हूँ। लास्ट 9 ट्रायल में लैंग्वेज इज मोस्ट इंपॉर्टेंट, एंड आउट ऑफ 9 ट्रायल 175 आर पार्टिसिपेंट्स, एंड इंक्लूडिंग दैट 98% ने हिंदी में कंसेंट अपना दिया है टू अंडरस्टैंड दैट कंसेंट इजीली। ये एक कंसेंट का एग्जाम्पल है। मानव जीवन की बेहतरी के लिए किए जाने वाले नैदानिक अनुसंधान में स्वैच्छिक भागीदारी के लिए आपकी रुचि के लिए हम आपका धन्यवाद करते हैं, क्योंकि आप इस अध्ययन के अभिन्न हिस्सा हैं। यह आपके लिए बहुत महत्वपूर्ण है कि अध्ययन की पूरी प्रक्रिया को समझें और सभी अध्ययन से संबंधित आवश्यकताओं का पालन करें, और इस दस्तावेज में अपने हस्ताक्षर या अंगूठा का निशान लगा के इस तरीके के कंप्यूटराइज्ड इन्फॉर्म्ड कंसेंट जो हमारे पास आते हैं मोस्ट ऑफ द ट्रायल्स में, उनको हम इजी तरीके से उसको हम अपनी लैंग्वेज में ढालें। जैसे क्लिनिकल रिसर्च एक शोध है जिसे हिंदी में नैदानिक अनुसंधान, इसमें विज्ञान का लाभ उठाकर मानव जीवन को बेहतर करने के लिए करना है। यह शोध सभ्य समाज की जरूरत है। अच्छे नए उपचार के लिए आपको दवा के इस अध्ययन में अपनी मर्जी से भाग लेने के लिए हम आमंत्रित करते हैं। इस तरीके के हम उसको कन्वर्ट करते हैं, उसको समझाते वक्त पूरी प्रक्रिया को समझिए और लास्ट अंत में अपना हस्ताक्षर या अंगूठे का निशान लगाकर इसका कॉपी आपको दी जाएगी। ये हम हर प्रोटोकॉल में हर इन्फॉर्म्ड कंसेंट को एक आसान हिंदी में उसको समझाने की कोशिश करते हैं। इसका रीजन क्या है? रीडिंग लेवल इज हाई। जो जितने भी कंसेंट प्रोसेस है 2016, 17, 18 में जो कंसेंट ये इन्फॉर्म्ड कंसेंट डॉक्यूमेंट मेरे पास आते थे, उनकी पेजेस थे 10, 11, 8, 9, 10, 11। आज इन्फॉर्म्ड कंसेंट जो कोवैक्स नोवैक्स का है या कोविड से रिलेटेड है 18, 19 पेजों का है। तो कंसेंट लेवल का जो वो है इंक्रीज हुआ है, लेंथ इंक्रीज हुई है, एंड उसकी रीडिंग लेवल इज हाई। ये जो रिसर्च बता रही है कि 11th ग्रेड और हायर की उसमें सेंटेंस यूज होते हैं टू अंडरस्टैंड द कंसेंट प्रोसेस। सो कंसेंट डॉक्यूमेंट लेंथ इज ऑल्सो इंक्रीज, उसका हायर वर्जन हुआ है, हायर लेवल ऑफ अंडरस्टैंडिंग, उसको कैसे समझा जाए। सो दीज आर द फ्यू पॉइंट्स फ्रॉम द वेरियस रिसर्चर्स की नेचर ऑफ रिसर्च 27% को समझ आया अकॉर्डिंग टू दिस रिसर्च। रिस्क कितनों को समझ आया, रिसर्च प्रोसीजर कितनों को समझ आया। ऐसे एक हमने रैंडमाइज कंट्रोल ट्रायल देखा, दिस मेटा एनालिसिस चेक किया जिसमें डिफरेंट कंपोनेंट ऑफ इन्फॉर्म्ड कंसेंट हमने चूज किए। नेचर ऑफ स्टडी, पर्पस ऑफ स्टडी, थेरेपेटिक मींस कंसेप्शन, रिस्क एंड साइड इफेक्ट, बेनिफिट। इसमें देखा गया मोस्ट ऑफ द 1/3 से 50% जो कंपोनेंट ऑफ इन्फॉर्म्ड कंसेंट हैं वो ट्रायल पार्टिसिपेंट को समझ नहीं आते। तो इसको इजी करके इजी वे में समझाना है और अंडरस्टैंड, उसको समझ में आना चाहिए। पेशेंट को हर पॉइंट को एम्फसाइज करना है हमें। तो इन्फॉर्म्ड कंसेंट प्रोसेस।
Dr. Balla,
Dr.
sorry to
Hira.
disturb you. Please speak in English because many
Yes.
of our participants don't know Hindi.
Okay, sir. Okay.
Okay, in English.
Okay. To understand participation in and the component of the informed consent, there are number of component of informed consent, and according to this meta-analysis, 1/3 to half of the participant unable to know or understand the various component of this informed consent. So informed consent process, this informed consent process should be draft the patient information sheet, including patient information sheet and ICF. These are two parts of informed consent document: patient information sheet and informed consent form, जिसमें वो in which he or she going to sign as soon as the protocol is final. Next point is obtain the approval of informed consent document, that is PIS and ICF from the ethics committee. Present consent document to participant, explain all the significant points of document, promote the subject to ask question and answer all the queries, refers to people's, he or she refers to people's on understanding the willingness and to participate in study, and it merely not about the signing of the form. It involves educating subjects and not just disclosing the information. And it is an ongoing interactive process. Consent process is an ongoing process and it does not end once the consent form is signed. And the information and clear understanding from the participant should be continued throughout the participation. So this is a prospective in nature, and throughout the participation, participant is informed. Ensure the readability of consent document. Write the document in the second person. Use appropriate font size. Use simple and non-technical language, as we discussed previous. Use commonly recognizable terms and measurement amounts. Offer explanation to terms that may be difficult to understand. Allow the subject time to discuss with family and friends. Meet the subject again. Ask the subject about open-ended question about the nature of study and participation. If the subject is willing, have him or her sign on the document. Give the subject copy of consent document. Check out what should be done before leaving the person who signed the consent. Review the consent document. Make sure all the sections have been correctly signed, dated by the participants or legal acceptable representative and the person obtaining consent. Many consent have more than one signature page. So participant or legal acceptable representative must personally sign and date the consent. Ensure that participation or legal acceptable representative has signed all appropriate section. Ensure the person obtaining the consent has signed all the appropriate section. Sign and date is very much important. So what are the various elements of patient information sheet? Patient information sheet must include the following points. The trial involved research, purpose of trial treatment, and the patient is randomized on which head, and the trial procedure to be followed, including invasive procedures. The approximate number of subjects which is involved in the trial. What are the subject responsibility? What are the those aspects of the trial that are experimental? Reasonable, foreseeable risk or inconvenience to the subject. And important is benefit. What benefit he or she will get? Alternative procedure. What is available to the subject and their important potential benefit as well as risk also. Treatment available. Anticipated prorated payment, if any, to the subject for participation in the trial. And voluntariness. The subject participation in the trial is always voluntary. He or she may refuse the participation or withdraw from the trial at any time without penalty or loss of benefit to which the subject is otherwise entitled. Next important thing, the monitor or auditor or institutional ethics committee or regulatory bodies will be granted direct access to the subject original medical records for verification of clinical trial procedure and or data without violating the confidentiality of the subject. This is very much important. If the result of the trial is published, the subject identity will remain confidential. And next point is, whenever there is a continuous process, this informed consent process is a continuous process. The subject or legally acceptable representative will inform in timely manner if information become available that may be relevant to the subject willingness to continue participation in trial. And next important thing is contact person. Whom to contact? The person to contact for further information regarding the trial and the rights of trial subject, and whom to contact in the event of trial related injury. He or she will contact to principal investigator, and ethics committee number is also there. Any foreseeable circumstances reason under which the subject participation in the trial may be terminated. This is also explained, and the expected duration of subject's participation in trial. Essential step for conducting informed consent. Obtain informed consent from each participant age more than 18 years. Parents plus child of age group 7 to 12 give the verbal assent. Parents or legally acceptable representative plus child of age 12 to less than 18 can written assent. So legally acceptable representative can include with both verbal assent and written assent. And this should be done before the start of any study. So legally acceptable representative, mother, father, brother, sister, all are legally acceptable representative for an individual or juridical or other body authorized under applicable law to consent on behalf of prospective subject to the subject's participation in a clinical trial. Who is impartial witness? This is very much important. Impartial witness is not a study team member, number one. An impartial witness is not a friend or a family member to the subject. This is very much clear. Impartial witness, it should not be a study nurse or co-investigator or any team member of the study. Impartial witness should not be from any family member for the study. Who will sign the consent form? Patient receiving the information and patient giving the information, both will sign. Who will sign the consent form? Patient receiving the information is study subject. If study subject is illiterate, legally acceptable representative will sign. In case of vulnerable population, legally acceptable representative will sign. Who will sign the consent form? If LRA is illiterate, we can take impartial witness. If subject is or LRA cannot understand the local or national or the English language, he or she cannot participate in that study. In case of special situations like informed consent in children, if less than minimal risk, one parent can sign the signature page. If more than minimal risk, both parents can sign. If child is more than 7 years of age, assent will be taken. 7 to less than 18, assent will be taken. 7 to 12, verbal assent, and 12 to 8 less than 18 is written assent. In emergency situation, prior to problem, we can take the consent from the patient, or if the prior consent is not taken, we can take the consent from legally acceptable representative. If LAR is not present, not available, as per protocol, as information informed to the ethical committee, we should be informed to the patient after the as soon as the to the patient after any problem, the situation over or emergency is over. Who support the application of basic principle of ethics in research? Our basic principle of ethics research is supported by ethics committee and informed consent. So please remember, informed consent is the only part of making research ethical. And this is informed consent is a legal form which help to maintain the principle of ethics in a research. Now comes to different types of consent. Till now, what I have told you is regarding the written informed consent. Regarding the AV consent is mandatory only in case of vulnerable population and with research on new chemical or new molecular entity. AV consent should be taken in a vulnerable population and the research on new chemical or new molecular entity. The condition with high resolution camera frame must include participant or legally acceptable representative, impartial witness, and PI. And requirement is the room free from any disturbance to ensure that the image is recognizable and audio is clearly audible. Audio consent is for leprosy and HIV patient. For e-consenting, electronic consenting, or previously we seen in the COVID problem, e-consenting is and remote monitoring is useful in taking consent during COVID pandemic. And this consenting is very much comprehensive discussion. And the document is this is conserved appropriately. And electronic formats such as text, graphic, audio, video, podcast, or interactive website to explain information related to study. We must take permission from ethics committee for electron material which we used. Method of documentation including electronic or digital signature. Method used to maintain the privacy of participant. And security for the information, data use policies at the research site. The electronic consent must contain all elements of informed consent in a language understandable by the participant. Electronic methods should not be used if participants, for any reason, indicate a lack of comfort with electronic media. Next is waiver of consent. The waiver of consent, research cannot practically be carried out without the waiver, and the waiver is scientifically justified. In case of retrospective studies, anonymized biological sample data, certain types of public health studies, surveillance program and program evaluation studies, or data available in public domain. Research during humanitarian and also in research during humanitarian emergencies, disasters, when the participant may not be in a position to give consent. Attempts should be made to obtain the participant consent at the earliest. Next is reconsenting when it is necessary. As directed by our ethics committee, depending on most recent approval, if there is any new information pertaining to study or based on change that may impact the participants' willingness to continue in the study, we go for the reconsenting. And for reconsenting, if we already taken the consent from LRA, legally acceptable representative, when the participant was enrolled using LAR, regain the cognitive ability to consent for themselves, she can consent by his own, no requirement of LAR. So we respect the basic principle of ethics in a clinical research, that is autonomy, beneficence, and justice. Autonomy, choose freely. Each person should be given respect, time, and opportunity to make his or her own decision. Protection should be given potentially vulnerable populations such as children, elderly, mentally ill patient, students, prisoners. Beneficence obligates the investigator to secure the well-being of subject and balance the risk and benefit and justice. All people are treated fairly. Its burden and benefits are shared equitably, and doctor-patient relationship to be maintained. Here are the few examples of informed consent document, which include participant information sheet and informed consent form. The first point is purpose. This is the purpose which is elaborated. This is regarding the rabies studies. Next is procedure. Procedure is well documented, and all the regarding all flowcharts to be understood easily by the patient or participant. Next is side effect and risk and discomfort to be explained. What side effects to be occurring during this likely to be caused during this study. Next is compensation for the study related injury. Insurance policy should be told to participant, or any related study related injury should be told to participant. Benefit, alternative, right to refuse, withdrawal of participation from the study. Sorry. And voluntariness and confidentiality. Benefit, alternatives, right to refuse from you or your child. Participation in the study is voluntary. All the points, all the elements of informed consent, it is shown here. If any new information is available, we can give the chance to reconsent. Any circumstance under which your participation may be terminated. If new information is available, what to do? And you may ask question, or you may have a question about the study. Contact number, detail, address of ethics committee and PI, principal investigator, is given, and we will give you the signed copy of consent form for this. This is the pictorial diagram of the assent form. It is the informed assent form regarding the rabies studies, which is easily understandable by children. This is the informed consent, and we have to write the screening number, participant's name, date of birth, sex, participation address, complete address, qualification, his student, service, self-employed, housewife, annual income, nominee, relation with the nominee, address of the nominee, contact details of the nominee, and this is the signature page. 6 points where we have to take the signature of adult or parent or legally acceptable representative. And this is the explanation, and this number first point also described, understood the information sheet dated. Information sheet is dated on which it mentioned in the first line. Next is the consent from the participant age more than 18 years of age. And this is the signature page for assent form. Parents or guardian or legally acceptable representative consent form. Sorry, consent form it is. And last is investigator or designee should sign here its name, signature, and date. With this, I conclude my presentation and summarize. Informed consent must be understandable. Informed consent is the only part of making research ethical. A long list of risks are not helpful. Need to synthesize information to make it meaningful. And we must include all the elements of informed consent. And who will consent is the important point here. who will consent regarding the impartial witness or regarding LAR, or the patient is participant itself. Thank you all for having me. Thank you, sir.
Thank you, Dr. Bala, for your crisp presentation. And some of the questions were raised during the presentation, and they have been answered by Dr. Bhagwat. I think that will do for that. Anything else somebody wants to ask? And I give over to Dr. Kamal.
Dr. Dakle, sir, if there are questions, then you can
Yes,
speak
sir.
on the behalf. Yeah.
Balla sir, how can we document the verbal assent? One question is in chat box.
Okay, sir. तो verbal assent में सर प्रायर इंफॉर्मेशन हमें एथिक्स कमेटी से लेनी पड़ेगी कि अगर 7 to 12 years of age है तो उसका फॉर्मेट हमें we have to make a format of that verbal assent.
And no, they are asking how can we document verbal assent after taking the verbal assent from the respective subject. That form has to be signed by his or her parents. If parents are not available, then LAR can sign that document. In that way, it can be documented. So next question is, what is minimal risk?
Minimal risk is, if we use the what minimal risk.
Actually, minimum risk is the risk that we can say that it is a routine activity, whatever the routine activity we carry out daily, and that is called as the routine, what we call it as a minimal risk. If the minimum risk is more than the routine activity, then it comes under the full review or whatever it may be. So minimum risk is the risk which can be attributed that during your day-to-day activity.
Yes, sir. If we give the injection to the patient and
Yes.
its routine activity is not hampered, then it goes to the minimal risk. If its routine activity is hampered, then it is going for the more than minimal risk, sir.
And specifically, if we define that risk, the minimum risk means that the probability and magnitude of the harm or discomfort anticipated in the research are not greater in and of themselves than those ordinarily encountered in daily life or during the performance of routine physical or psychological examination or the test. This is the specific definition of minimal risk. Means day-to-day activity जो अपन करते हैं उसमें अगर जो active risk अपन accept करते हैं, that is called as minimal risk. If it is more than that, then it does not come under the minimal risk. The last question we can take: child. Can one study participant be impartial witness for another study participant?
Sir, what's the question, sir? I could not understand.
Question is, can one study participant be impartial witness for another study participant? Means if there are two study and one study participant enrolled in one study, can he become the impartial witness for the another study?
Partial witness for the another study. Both are simultaneously occur at one site.
No. It is regarding the impartial witness. The answer is that even one person is included in one trial, he can become the impartial witness for the another trial, provided he should not be the relative of that participant and he should not be the team member of that protocol or the study team. Then if that two conditions are satisfied, then that person become the what we can say as a eligible for the impartial witness.
Okay, sir.
So with that, we will conclude today's session. Before concluding the session, I request Dr. Kamal sir to say a few words on NPT. Over to Kamal sir.
Thank you, sir.
Good afternoon, all. On behalf of our Honorable President, Dr. Padamja Uday Kumar, I am having some announcement to be passed. that I just want to convey that apart from the registration, online registration, the participation in the online activity is also mandatory for obtaining the certificate of GCP workshop. And we are continuously working behind the scene also with National Association of Pharmacology and Therapeutics. Our journal is coming probably next month. So those who are willing to send their articles, they can send. We will be sending the email details for sending all these articles on the WhatsApp and email also. And our newsletter that is in the name of the RX Factory is also coming next month. Those postgraduate students can also participate in RX Factor activities, and they can send their articles. This time, along with the articles, we will be printing their photographs on the RX Factor. So with these words, I just declare the conclusion of this session. Thank you all for joining us. And thank you from National Association of Pharmacology and Therapeutics.
Thank you, sir. Thank you so much.
So we will meet again on 23rd March at 3 p.m. Thank you all.
Thank you, sir.
Bye.
Mm.